Hi, my name is Jesse Poganik, and I'm happy to provide this video abstract to give an overview of our recent perspective in Bioessays titled Getting the Message, Native Reactive Electrophiles Pass 2 out of 3 Thresholds to be Bona Fide Signaling Mediators. The focus of our perspective is on reactive electrophilic species, or RES, and a role for RES as true signaling mediators in cells. In this manuscript, we lay out 3 thresholds we feel must be passed by RES in order to cement their role as signaling mediators. The first threshold is one of suitability and feasibility, or put another way, can RES modifications constitute a bona fide post-translational regulatory mechanism? This threshold has been passed over the years by careful pioneering work in the redox signaling field. Early data were somewhat confounding, but clearly hinted at a role for prostaglandin RES as cell signaling mediators. More recently, groundbreaking proteomics methods have allowed us to rank the susceptibility of proteins to RES modification. Although there are some important limitations to these techniques, such as their reliance on external administration of large concentrations of RES, the ability of these methods to identify subsets of proteins that are uniquely reactive with RES makes a strong case for their role in cell signaling. Because the field has further been able to link modification of these identified proteins to changes in cell signaling, threshold 1 has been crossed. However, the nature of the leading RES proteomics methods requires proteins to be modified at high occupancy to be scored as a hit, whereas canonical cell signaling mechanisms likely do not require high occupancy to function. Addressing this aspect of RES signaling is where threshold 2 comes in. The second threshold we propose is one of specificity and sufficiency, and as I alluded to earlier, addresses whether low occupancy RES modifications can drive dominant signaling responses. The methods we discussed that led the field to cross threshold 1 are not particularly well suited to addressing the question of threshold 2 because of their reliance on global administration of RES to cells or lysates, wherein manifold protein targets are hit by the RES. With these setups, effects of discrete modifications of specific proteins at low occupancy cannot be addressed against an otherwise unperturbed proteome. To overcome this limitation, our lab pioneered the targetable reactive electrophiles and oxidants, or TREX system. TREX utilizes a pseudo-intramolecular reaction to deliver a maximally stoichiometric amount of a given RES to a given protein of interest. Because the delivery we achieve with TREX is necessarily substoichiometric to the protein of interest, we engender low occupancy RES modifications. Importantly, however, we've shown that TREX RES delivery to a specific protein largely does not perturb the signaling dynamics of the remainder of the proteome. With TREX, we've been able to identify a number of proteins which we refer to as kinetically privileged first responders to the RES under study. Most importantly, in terms of crossing threshold 2, low occupancy RES modification of these proteins is sufficient to drive measurable downstream signaling responses. The third most challenging threshold is one of functionality and physiology. Can RES modifications and resulting downstream signaling operate under physiological conditions? Although the compendium of the evidence gathered over the years, including insights gained by TREX and proteomic methods like ADPP, collectively suggests that endogenous RES signaling is possible, we do not yet have direct evidence that this is the case. In this section, we propose some approaches that may be able to prove that endogenous signaling can occur. We also speculate on how, from an evolutionary standpoint, RES signaling may have come about, and relate these ideas to potential future lines of investigation that may be able to unequivocally prove that RES signaling is a bona fide cellular signaling mechanism. Finally, it's important to note that even as the field continues to work to traverse threshold 3, identification of RES-responsive proteins holds great promise for drug development, especially as we see a resurgence of covalent inhibitors targeting kinetically privileged cysteines. In summary, our bioessays perspective lays out 3 thresholds that RES must pass to be considered true signaling mediators in cells. We show how the first 2 thresholds have been crossed by the chemical biology community, and lay out some possible ways the third threshold can be crossed. We hope that you enjoy the paper, and we hope that it will inspire more scientists to enter the exciting and intellectually stimulating field of electrophile signaling.